What's up guys, Dr. Goodin here, and in this video we'll be talking about the structure and function of muscle tissue. Okay, let's roll that intro. Before you can really understand training physiology and programming and periodization, you have to have at least a basic understanding of muscle physiology. A basic understanding of muscle physiology sets you up to better understand some of the principles by which we train, some of the neuromuscular mechanisms that we can harness or potentiate during training, and it helps us to make logical training decisions based on anatomy and physiology of the athletes that we are training. So today we're going to look at a basic overview of the structure and function of muscle tissue. Okay, so let's dive right in. Okay, this comes from Chapter 1 of the Essentials of Strength Training and Conditioning textbook, put out by the National Strength and Conditioning Association, authored by Drs. Haff and Triplett. Okay, so the muscular system. We want to talk about both the macrostructure and the microstructure, so big picture and small picture of muscle tissue. What's happening at a whole muscle level as well as what's happening at the cellular level in muscle. We're not going to go too deep, I'll save that for exercise physiology, but we do need to know the basic overview before we move forward. So each skeletal muscle is in itself an organ that contains primarily muscle tissue, but also connective tissue, nerves, and blood vessels, right? So this tissue needs to be innervated, it needs to have blood flow for oxygen, obviously, and then also to remove waste products, and it needs connective tissue to kind of hold it all together and give it a resiliency. Now, if we check out this diagram, we see three layers of connective tissue. The outermost layer is the epimysium, and that's going to run along the perimeter of this muscle. Deep to that, we have the perimysium running in between these individual muscle fascicles. Okay, fasciculus is the singular version of that word fascicles, and literally the fasciculi, that just means a bundle of sticks in, I don't know, Latin or Greek or something. So it looks like a bundle of sticks, right? So early anatomists thought, oh, look at these long muscle fibers. It looks like this bundle of sticks. Let's call it a fasciculus. Now within this fasciculus, we have the endomysium running between individual muscle fibers. Okay, and so those are your three levels of connective tissue. Now, it's important to remember that all of these levels of connective tissue are actually continuous with the muscle tendon. So when tension is placed on any individual muscle fiber, whether by contraction, actively, or by passive stretch, that tension runs through the length of the muscle. And it's what allows the muscle to develop tension from one point of origin to its point of insertion. Now, in this fasciculus, we have these individual muscle fibers. Okay, so here's that single muscle fiber coming out of the fasciculus, and you can see that it's multinucleic, right? So we have all of these multiple myonuclei or muscle cell nuclei. And the reason why it has so many nuclei is because it's such a long cell. It has a large domain which each of these nuclei must control. And so with a single nucleus, this cell could not manage all of the cellular functions going on. And so muscle cells are very unique in that they have multiple nuclei. And at some point when we talk about hypertrophy, we'll actually talk about how satellite cells donate nuclei to expanding muscle cells in order to increase the number of nuclei and expand the volume of that muscle fiber. So as you enlarge your muscles through the process of hypertrophy, the enlargement of a muscle cell, you can actually gain more myonuclei. So we see that inside of the single muscle cell, we have sarcoplasm, which is analogous to cytoplasm of a cell. And we have these myofibrils. These are the contractile strands within the cell that are themselves composed of myofilaments. And the two myofilaments are actin and myosin. Oops. actin and myosin. So actin filaments are thin and they're woven together with strands of actin, and myosin filaments are thick and they have these globular heads that latch on to the actin filaments. Now, I mentioned that we have not only muscle tissue, but also nerves and blood vessels and connective tissue. So let's talk about the functional unit of the muscle, which is actually comprised of both nerve tissue and muscle tissue, and it's called the motor unit. So the motor unit is the contractile unit of muscle tissue. A single motor unit consists of the motor neuron, also called the alpha motor neuron, and all of the muscle fibers that it innervates. Okay, so here we see a picture of the alpha motor neuron. Alpha motor neuron, and we can see that it is innervating multiple muscle fibers within this muscle cell. So each of these little neuromuscular junctions is representing a single fiber that this alpha motor neuron is innervating. There are typically several hundred muscle fibers in a single motor unit, but that can vary. The motor units of your eye, for instance, contain only a handful of muscle fibers per motor unit, while the muscles of your quadriceps, those motor units contain hundreds of muscle fibers per motor unit. It depends on how finely we need to grade our force output of these muscles. Now, zooming into a muscle cell, we see some important distinguishing characteristics that set muscle cells apart from the other cells of your body. We can see the mitochondria floating around, that's similar to other cells, but we have what are called T-tubules or transverse tubules, and these openings in the cell membrane actually allow an action potential to infiltrate and spread across this long muscle cell very, very rapidly, so that you can have a more synchronous contraction of all of the contractile elements within the muscle cell. We also have what's called the sarcoplasmic reticulum. So in the sarcoplasmic reticulum is where the muscle cell stores and releases calcium. Calcium is super important because it's a second messenger, the release of which is signaled during or after an action potential in order to travel into the contractile elements and allow actin and myosin to bind together, as we'll see in a moment. The membrane of a muscle cell is called a sarcolemma instead of a plasmalemma. And then, of course, we see the myofibrils that we talked about previously. So the main function of muscle tissue is to contract and to generate tension between two different points on the skeletal system to create movement and motion. The way that muscle tissue accomplishes this is through what we call the sliding filament theory. This slide here shows the multiple levels of detail through which we can observe the sliding filament theory. Okay, so let's zoom further into this image and go through step by step. So first we see the whole muscle here. And if we zoom in on the fasciculus here, and then even further into an individual muscle fiber there, and finally to the level of the myofibril, then we see within these myofibrils what are called sarcomeres. And a sarcomere is the contractile unit of the muscle cell. And they run from Z line to Z line, and in between those Z lines is the boundary of the sarcomere. So we see here the Z lines, and so what we're looking at is actually two sarcomeres in this image. And we notice that sarcomeres have this striped or striated appearance, and that is caused by the overlapping regions of the different contractile elements, actin and myosin. So remember, myosin are the thick filaments, and they're shown down here, the pink ones, and the actin are those lighter colored filaments. So going back to our pictures of the sarcomere, then we have the M line, which is the anchor point for the myosin, easy to remember, M for M line and for myosin. And we also have what's called the H zone, and the H zone is where you only see myosin, okay, so it's going to be pinker. Then the A band is the region within which those myosin filaments extend from end to end. The A band is where you have myosin filaments. The I band is where you have only actin filaments, but no overlap with the myosin filaments. So the I band is lighter in color and it's only the actin filaments, and in between there you have your Z lines. Now, when we look at the myosin filaments close up, we see that they have these globular heads. And it is these heads that complete what's known as the power stroke, right? The latching and then power stroking and then breaking and then relatching on the actin filaments to actually complete the contraction and the shortening of a sarcomere. And in between those, we have the braided actin filaments, and they're actually covered with this troponin-tropomyosin complex that you see here, which blocks the binding sites for the myosin heads until calcium enters the cell. So remember, we talked about how the sarcoplasmic reticulum stores calcium and then releases calcium when an action potential infiltrates the T-tubules on the muscle cell and allows that action potential to then allow this release of calcium into the myofibrils, which then causes the troponin-tropomyosin complex to move and allow the myosin heads to bind on the actin. And then in the presence of ATP, which is the body's energy currency, then we have the power stroke and the relatching occurring. Okay, so fairly complex. However, if we look at it from this big picture view, zooming in from the whole muscle down into the individual sarcomere, I think it gives us a better understanding of what's actually happening during a muscular contraction as you develop tension in the muscle down to the sarcomere vantage point. And this has implications into how we understand things like the stretch shortening cycle, or different types of muscle contraction, eccentric, concentric, and isometric, or into how we program rest periods based on which energy system that we're using. So very important to understand this fundamental underlying basis of muscle physiology. So the key point here is that it's the discharge of an action potential from a motor nerve that signals the release of calcium from the sarcoplasmic reticulum into the myofibril, causing tension to develop. So that was a very succinct way of explaining in one sentence everything that we've just talked about, from the alpha motor neuron down to the individual contraction of a sarcomere. That's the path that it takes. Now, to summarize the sliding filament theory of muscular contraction, we would say that actin filaments at each end of the sarcomere, anchored to those Z lines, slide inward on the myosin filaments because of the globular myosin heads performing the power stroke, and that pulls the Z lines toward the center of the sarcomere and thus shortening the muscle fiber. So when we have all of these different sarcomeres, and they don't always shorten all at once within a muscle, but you have various sarcomeres shortening and pulling, and remember they're bound by 3 different layers of muscle fascia. So as these sarcomeres shorten, that fascia is generating tension, and the tension, because it's continuous with the muscle tendons, anchored to your skeletal system, that generates tension and force on your bones. And that force creates leverage on the skeletal system and allows you to move your body and external objects. Okay, so we have the big picture view of muscles moving on bones and the very microscopic view of what's happening at the sarcomere level. Now, let's take another look at it, going step by step through the contraction of a myofibril. So in this first phase, we'll just call this A, this is a stretched muscle, and the I bands and H zones are elongated because there's not very much overlap between the myosin and actin filaments. Okay, that's the overlap. So the I band, which is only actin filaments, and the A band, which is and the H zone, which is only myosin filaments, those are stretched out. There's also a low force potential. Why do you think there would be a low force potential? Well, it's because it's the globular heads of these myosin filaments acting on the actin filaments that produces force within a muscle. The contractile force of a muscle can be really reduced down to the number of myosin and actin binding sites at any given moment. And so if there's very low potential for binding between the myosin and actin filaments, then there's low potential for muscle force. Now, in The second phase of contraction, this is when muscle is contracting, right? Partially. It's not fully contracted to its shortest length, but maybe it's at its most optimal length for force production, and that's because, again, we have all of these cross-bridge potentials. Now there's a much greater potential for cross bridges to happen because all of these myosin globular heads can theoretically be bound to the actin in performing a power stroke. Okay, so this is where we get into optimal length tension relationships. You'll notice that muscle performs best or is strongest at certain lengths, and it's different for each muscle depending on the orientation of its fibers and the fiber typing, etc., and then also depending on its origin and insertion and what type of lever it's creating. But for muscle tissue itself, just for the force of a single muscle fiber, there is some sort of happy medium where there is the maximum number of myosin and actin cross bridges that can form. And then finally, once muscle is completely shortened, there is again a low force potential due to the reduced cross bridge potential. And we see here that there's actually an overlapping of the actin, and the myosin is actually running into the Z lines over here. And so, for instance, if you've ever tried to do a bicep curl and you hold it at the top in that maximally shortened state, there's not a lot of force you can produce there because your muscle's already shortened. Or if you have ever done any type of like hamstring curl or a standing, or a standing knee bend where you flex your knee and bring your heel up to your butt and you try to hold that for as long as you can, those muscles are maximally shortened and they'll begin to cramp because they're actually running into each other. The sarcomeres are physically out of room. They can't go any further, and therefore the muscle is maximally shortened. Okay, so let's recap really quick the phases of the sliding filament theory of muscular contraction. First, we have the resting phase, right? This is when there's no action potential, no calcium has been released from the SR. And then we have the excitation contraction coupling phase. This is when an action potential has traveled down the alpha motor neuron, it has infiltrated the T-tubules, released calcium from the sarcoplasmic reticulum, and the calcium is now going into the myofibrils. We have the contraction phase, and this is when the coupling of the actin and myosin is happening. Cross bridges are forming, ATP is present and allowing the cycling of the myosin head. And calcium's in there as well, removing the troponin tropomyosin complex from the binding sites to allow this to happen. Okay, and fourth, we have the recharge phase, and this is when we require that ATP to release the myosin head so that it can engage again and perform another power stroke. And finally, the relaxation phase. The signal has stopped coming down the axon of the alpha motor neuron, and so now calcium is being taken back up into the sarcoplasmic reticulum, and that covers the binding sites, and so the myosin heads are no longer able to bind to the actin, and therefore tension is reduced, force is reduced within the muscle, and it relaxes. Okay, so key take-home point here that I've already said, but I'll reiterate. The number of cross bridges that are formed between actin and myosin at any instant dictates the force production of a muscle. So actin and myosin cross bridges dictates force production. The more potential you have for actin and myosin to bind, the more force. So that means a larger muscle, which has more contractile elements shoved into each of those muscle fibers, will be a stronger muscle. Okay, all other things considered. Now, yes, there are things like rate coding and neuromuscular factors that we're going to talk about in the next lecture, but all other things considered, the larger the muscle, the more opportunities for actin and myosin to bind, and therefore the stronger that muscle is, the more force it can produce. Another key point, calcium and ATP are absolutely necessary for cross-bridge cycling with actin and myosin filaments. So you need calcium in order to physically allow the contraction to happen, and you need ATP to allow those globular heads to detach so that they can complete another cycle. Okay, and that was a basic overview of the sliding filament theory of muscle contraction. I hope that was helpful to you. In the next video, we'll talk about the neuromuscular system, factors related to recruitment and to motor units. So I'll catch you guys on the next video. But until then, move well, live well, keep teaching other people to do the same. This is Dr. Goodin. Thanks for watching.